The BioClock Studio Winter 2020 presents the Circadian Transcriptomics Dry Lab Tutorial Part 3 of 4, Differential Expression and Circadian Analysis. In the previous video, we discussed the process of RNA sequencing and normalizing sequencing read counts. This video will discuss how to analyze RNA sequencing data to discover the transcriptional basis of phenotypic differences in circadian biology. To identify the genes that may underlie circadian phenotypes, we employ differential expression analysis and circadian analysis. Differential expression refers to the comparison of changes in gene expression across two conditions, such as differing genotypes, tissue types, or diets. Say we observe an obese phenotype that differs among groups of mice depending on their feeding condition, such as ad libitum versus time-restricted feeding, or TRF. In order to figure out which transcripts are related to this phenotypic difference, one approach is to specifically look at genes that are known to be associated with lipid metabolism pathways. This is the manual curation method. A computational analysis can compare expression of these genes between feeding condition samples. Any genes that are expressed at different levels between the two conditions, which are said to be differentially expressed, can be further examined in future experiments to test their impacts on the phenotype. The general workflow researchers use to determine the complete set of genes that are differentially expressed between two samples is as follows. First, the counted and normalized data from the RNA sequencing for the samples to be compared are entered using software specifically designed to compute differential expression, such as DESeq2. Next, DESeq2 makes pairwise comparisons between the samples and indicates changes in the expression of individual genes. In our example, we compare sample 1, ad lib mice, to sample 2, TRF mice. DESeq2 will compare the expression levels for each gene under the two conditions and then annotate the data table with values for fold change and p-value. The fold change indicates the factor by which gene expression changes. For example, gene D gene expression levels are 60 and 100 for samples 1 and 2, respectively. This means that there is nearly a twofold difference in the level of expression between the two conditions. A negative fold change signifies a reduction in gene expression in sample 2 versus sample 1. The p-value reports the probability that this difference in gene expression is the result of random chance. The log of these numbers is calculated to better visualize the difference in gene expression. In the third step, the calculations made by DESeq2 are used to determine which genes are differentially expressed. The standard is to consider a fold change greater than 2 because a twofold change or more is likely to have biological effects, and a p-value less than 0.05 reflects an acceptably low 5% probability of obtaining these results by random chance. We have highlighted the fold changes that are less than 2 in pink and the p-values that are greater than 0.05 in yellow. We are unable to conclude that these genes are differentially expressed, so they will be filtered out of the dataset. After this step, the significant gene list is considered to be an experimental result and can be investigated further through a concept called gene ontology. In addition to differential expression analysis, circadian analysis can be applied to discover the complete set of rhythmically expressed genes and compare which genes are rhythmic under different conditions. For instance, it is possible to determine which genes are rhythmic in the liver of wild-type mice, but not BMAL1 knockout mice. One algorithm used to conduct circadian analysis is called MetaCycle. MetaCycle starts with a data table, which contains gene expression level data for each gene in a sample at each time point as shown here. For each gene, the expression levels are plotted for each time point. MetaCycle tests whether individual genes are rhythmically expressed by fitting the gene expression data plotted as expression value for each gene at each time point of sampling to a cosine curve that represents an idealized cycle. In order for the program to do this, it is necessary to set parameters for the period lengths expected to be observed if the data oscillate. Because circadian rhythms should be close to 24 hours, This analysis looks only for oscillations in gene expression with period lengths between 20 and 28 hours. After the parameters have been set, MetaCycle will calculate the amplitude, period length called tau, and relative phase. Also reported is a false discovery rate as a p-value for the rhythm in the expression of each gene. The false discovery rate is a statistical metric that indicates how well the data match a cosine function. These metrics are annotated onto the dataset in new columns for every gene, and non-rhythmic genes are filtered out. These rhythmic gene expression data are compared from different samples to determine which genes are rhythmic under different conditions. Researchers use a bioinformatics approach called gene ontology to address the following question: which cellular processes are showing up as being particularly relevant in the analysis under study? Gene ontology helps to connect differentially or circadian expressed genes to a phenotype from a bottom-up approach. The general workflow of gene ontology is as follows. First, the list of genes of interest that was obtained through differential or circadian expression analysis is entered into a gene ontology enrichment analysis tool, such as David or Metascape. Second, the tool will compare this list of genes to lists of genes that are grouped into specific biological processes. These functional groupings of genes are stored within gene ontology databases such as GO or GO, Reactome, and the Kyoto Encyclopedia of Genes and Genomes, called KEG for short, which are libraries of gene pathway associations that have been curated using the scientific literature. The mathematical test performed to make these comparisons is called a hypergeometric test. A Venn diagram is a nice visual representation of the test. As an example, if the gene ontology database shows that genes C, E, G, and I are associated with protein metabolism, and if genes C, E, G, and I are among the genes that are differentially expressed, what is the likelihood that their shared role in protein metabolism is why they were discovered together rather than just by random chance? If protein metabolism is different between the two conditions under study, we would predict that genes involved in protein metabolism would show up in the list of differentially expressed genes more frequently than in the list of all expressed genes. In other words, the list would be enriched for genes involved in protein metabolism. The gene ontology enrichment analysis outputs several metrics, and two important metrics are the fold enrichment for each pathway in the gene list being queried and the p-value. Lastly, the results can be visualized using a bar graph to plot the enrichment score, usually shown as the negative log base 10 of the p-value for each pathway that is overrepresented in a gene set. Together, the tools and methods we have discussed allow scientists to bridge the gap between a genetic or environmental perturbation and an observed phenotype. Through differential expression analysis and circadian analysis, it is possible to discover the sets of genes that display changes in their steady state or circadian expression patterns in response to clock gene mutations and/or TRF. Gene ontology analysis can then curate the repertoire of biological and physiological processes that are known to be regulated by the genes found in the RNASeq analysis and point to groups of genes that might be responsible for the observed phenotypes. In the final video of this series, we will discuss the various ways in which scientists can visualize their results.